Hi, everybody. Welcome to chapter 25. In this chapter, we're going to talk about microbes in our environment, as well as introducing this concept of One Health. These are our learning objectives for the chapter, so keep up with that as we're covering the content in the lecture. To start off with, this is introducing this idea of microbial ecology, which is a study of microbes in their natural habitats. We don't always just study them in petri dishes in labs. Sometimes we need to know a little bit more about how they interact with their environments naturally in the world. So we have gathered data from observing them in the environments as well as by gathering genetic content from them in the wild and using that to study them. Microbes can be important in many of our biogeochemical cycles. When we talk about the carbon cycle or the nitrogen cycle, microbes are involved in many of that stuff. It's not as simple as just plants and animals and how they affect things, but that microbes are involved in how we are breaking down matter and reintroducing our elements back into our environment, and then how those will be reabsorbed by other organisms. Starting off with our very basic carbon cycle and how that's being cycled in our environment. Carbon's obviously the most fundamental atom in all biomolecules. All organisms are made of organic compounds that consist of a carbon-hydrogen backbone. So carbon is very fundamental in this. A lot of biomass is basically made up of carbon. When we talked about in chapter 9, CHOMPs, the carbon, the hydrogen, the oxygen, the nitrogen, the phosphorus, the sulfur, carbon came first because it is the most prevalent, and that the CHOMPs is listed in the prevalence of those elements basically within living life. So carbon is the most essential. It exists predominantly in a mineral state in the environment around us, and then certain microbes and certain organisms have basically developed a way to turn inorganic carbon into organic bioavailable versions of it. This chart basically summarizes the gist of the carbon cycle. And in this, we can see that respiration, this literally means like aerobic respiration. We are exhaling carbon dioxide. So that's coming from basically an organism source. That could be microbes that are going through aerobic respiration and making carbon dioxide as a product of that, or it could be actual like macroorganisms like animals, for example. Then we know that carbon dioxide that's free in the atmosphere can be fixed by our photosynthetic organisms. This is not just plants. This could be algae and cyanobacteria as well. Then taking inorganic carbon dioxide and fixing it into organic molecules. So that's then can be used by consumers, including animals, protozoa, helminths, and all of that can be cycled through the organisms in our environment. And saprobes, which are organisms that basically degrade dead things, right? So we learned about that in the previous chapter. The saprobes will also decompose anything that has died and then release all of that carbon dioxide back into the atmosphere. Carbon dioxide is also released through the atmosphere through combustion. That could be combustion from like normal volcanic activity or burning, fires, wildfires, but also could be industrial burning as well, which can release that stuff into the atmosphere. Carbon dioxide buildup in our atmosphere has contributed significantly to the change of our environment. So if you look at this chart here, just from 1960 up into 2020, this is talking about the carbon dioxide in parts per million in our atmosphere. And let me tell you, I mean, it was basically flatlined before, our industrial revolution. The industrial revolution has significantly spiked the amount of output of carbon dioxide into our environment. And carbon dioxide is a greenhouse gas. And what that means is that it traps heat from the sun basically within the planet. And so it continues to heat up and heat up and heat up rather than releasing some of that heat back out into space and things like that. So it creates a blanket sort of around the world. But carbon dioxide is not the worst offender. The worst offender is methane. And so methane is actually a gas that is produced by methanogens. And so these are organisms that live typically in anaerobic systems, often in the gut of organisms like us or cattle, stuff like that. So it's actually more potent, almost 20 times as potent as carbon dioxide at trapping that heat in our atmosphere from the sun and helping engage in global warming. So it's a major producer of that. So we see about 20% of the global methane production coming from cattle in the livestock industry, not just cattle, but any livestock. So sheep, cattle, goats, all of those livestock releasing methane into the atmosphere as part of the agriculture industry. And it's a huge contributor to global warming. Moving on to the nitrogen cycle, how do microbes contribute to this? They're actually probably the most important part of the nitrogen cycle in nature. Nitrogen gas in the form of N2 is 79% of our atmosphere, but it's not usable by me or you or by plants. So how do we get nitrogen into our system? First things first, when we talked about all of those elements that were important for life, and we said chomps with an N, nitrogen is here. Why do we need nitrogen? We know we need carbon because organic molecules, but why do we need nitrogen? What major macromolecules have nitrogen in them? Well, proteins do. They are made up of amino acids. Amino acids, amino literally means an NH2 group. This is nitrogen. So proteins with amino acids need to have nitrogen. All right. So what else needs nitrogen? Nucleic acids. Nucleic acids like DNA and RNA, deoxyribonucleic acid, and RNA, ribonucleic acid. What's the difference between DNA and RNA? Deoxyribo and ribo, an OH group on that ribose sugar. Deoxy is lacking it. It's been deoxied, and ribose has it. Okay, so that's the difference. Don't forget it because you know it's going to come up. All right, so that's our reminder about that. But all DNA and RNA are, is there a 5-carbon sugar, ribose or deoxyribose, 5-carbon sugar, pentose is what we call those, okay? That's attached to a phosphate, which is negatively charged, which is why DNA has a negative charge. RNA does too, okay? And then it's also attached to a nitrogenous base. This is what makes it A, T, C, or G in the case of DNA. Or in RNA, A, U, C, or G. That's a nitrogenous base. So we can't have protein, and we can't have DNA, and we can't have RNA. Ribosomes are made up of ribosomal RNA. You can't have ribosomes. You can't have anything unless you have nitrogen. And of course, all of these nitrogenous bases and all this stuff too, they need carbon and hydrogen and oxygen also, but they need nitrogen too. So we have to have nitrogen as a major building block. So that's why we need it. How do we incorporate it into those bigger molecules? So this relies on taking, literally at some point, we had to take nitrogen gas and incorporate it into these bases somehow. And so how does that happen? It happens through nitrogen fixation, ammonification, denitrification, and nitrification. Yes, I underlined them in a weird order, but yes. Okay, so nitrogen fixation, ammonification, nitrification, and denitrification. Tried to read them in order that one time. Okay, so this basically describes how the organisms are going to be converting from N2 into nitrates and making ammonia in the middle of it and all of that. So a lot of this can basically have to deal with dealing with nitrates, which is NO3, and going from NO2 to NO3 or NO3 to NO2. Again, this would be a reduction process of going from NO2 and then potentially going to NO or N2 or ammonia, which is NH4 and all that. So ammonium ion would be NH4. So you don't need to know the specifics of those chemical reactions, but you do need to know that these 4 processes are important in microbes basically converting nitrogen into usable forms. And plants can't do a lot of this. They can't use some of this nitrogen in the form that it's in. It can only use it in certain forms. And so microbes converting it from one form to another form allows the plants to use different forms. Okay, so higher plants utilize nitrate versions, and that this is basically going to be made by the bacteria for the plants. Without the bacteria doing this, the plants can't use it. And so this is a huge, huge thing that goes on in the soil. Because in the soil, we have bacteria that basically attach to the roots of plants and then do this for the plants. Basically, convert the nitrogen that's in the environment and in the soil into a usable form for the plants. This is a huge issue, probably bigger than you realize, because right now, with global warming going on, it's too hot for some of the bacteria to survive. Even though the plants could technically maybe live in some of these zones where it's super hot, if it's too hot for the bacteria or the bacteria don't have enough water, then they can't convert this into a usable nitrogen form and the plants can't grow. And then the people that rely on those plants for crops could starve. And this is what we're seeing actually happening in our environment right now actively with this global warming crisis. Moving on to water, understanding how water is basically how it exists on our planet. So we might have most of the planet consisting of water, but a very, very small portion of it is actually drinkable water. And so, subset of all of the water, most of the water on the earth is a saline version of the water, what we see in the oceans. 97% of all water on the planet is salt water in the ocean. That's not drinkable. Only 3% of it is fresh water. Of all of that fresh water, we have 68% of it in ice caps and glaciers. We have some of it in groundwater. We have some of it in other category, some of it in surface water, for example. And so of all of the usable kind of groundwater water sources and that other category where we usually pull our water from for drinking, most of our water that is involved in drinking coming from lakes and swamps and rivers and aquifers and things like that. It's very small surface. So very small amount of water that's available on the surface for us for consumption. We can convert some of these other sources into usable versions, but it just takes more work. Most water that falls on land will also be either returned to the atmosphere or used by plants. And so it's pretty amazing that a lot of this water is actually maintained in a usable, like that we're able to sustain life when a very small portion of water on the planet is in a usable format for us. So pretty interesting stuff. Microbes can live in the water, but there's a huge problem with our water environments on this planet as well. Because why? Because pollution. So the plastosphere refers to all the plastic that's basically existing in our environment and in our ecosystems right now. 88% of the ocean surface is coated with microplastics. Whether you see them or not, 88% of the ocean is covered with microplastics. And the likelihood of us being able to clean that in the immediate future is slim to none. And so we're looking for different ways to try to have maybe microbes be engineered to try to eat up that plastic, find ways to break it down. But in the meantime, some of that plastic creates sort of reefs in the ocean that are sort of like little islands where microbes can like create biofilms and new communities where they don't belong. It can block sunlight beneath them and actually disrupt the ecosystems that are anywhere beneath them with that needed sunlight that's now being blocked, or allowing like certain plankton that normally would live there can no longer live because the microbes are taking up all the resources and etc. So this is a huge issue for our water biomes and the effect of plastics and other sort of pollutants in those areas affecting the ecosystem overall. In particular for this course, talking about the microbe effects and then how those microbes can affect our ecosystem. For our drinking water sources, it's really important that we be aware that if you drink any groundwater, that there could be microbes in it. In fact, if there's a space for microbes to be, they're probably living there. Most of our drinking water comes from rivers, aquifers, and springs. So aquifers are basically like groundwater sources. And we usually have to process our water in order to make it safe for consumption. So we need to understand a little bit about how fresh water is processed to make it safe for consumption. So we would start off with our water source, which would be our lake or our river or whatever. In the case of maybe here in Oklahoma City, maybe the North Canadian River that flows into Lake Overholser. So maybe it's something like that. And then we usually have a step of coagulation. A chemical is added to make large particles settle out, and then we allow for sedimentation. Sometimes that sedimentation can be from large particles like dirt, debris, and stuff like that. And in fact, that's what Lake Overholser is. And that's also what Lake Hefner is. Both of them serve as sort of sedimentation points for large matter. I'm not saying there has to be chemicals added at that point, but at this point, we are trying to help things settle out and then letting that cleaner water that's up at the surface flow on to the next stage. So this serves as reservoirs, but also a sedimentation step. And we can add certain chemicals to help with that sedimentation process. Then we can filter out that top water, that cleaner water. Once the larger particles have settled, we can filter that top water and then disinfect that filtered water with UV treatment as well as chlorine-based chemical treatments. And then that is what actually can be stored, and that's what's actually transported to your house. But keep in mind, do you think that the pipes on the way to your house or the pipes that are in your house that lead to your faucet, do you think those pipes are sterile? Okay, just saying. We have to treat wastewater differently. This is poop water, okay? Pee and poop water, brown water, not gray water, which is from your sinks, brown water, okay? Sewage. So we have to treat sewage differently than drinking water because we know it's contaminated with microbes that can make you sick for a fact. So with wastewater, the sewage that is in that was from humans or animals or anything like that, it can contain all sorts of microorganisms and debris and chemicals and things like that that can get you sick. And if this is just dumped into our environment, then we could be just reintroducing it into our bodies and it could make us sick. In fact, this is one of the ways that we kind of define what makes a developed country versus an underdeveloped country is their ability to maintain hygiene related to their sewage practices. And so there's different stages to treating sewage. The first stage, the primary stage, we're separating out large matter. Yes, this could literally be plastic bottles and actual physical things like that, but we're also talking about solid waste coming from feces and things like that. The secondary stage allows biodegradation by our microbes. They're going to break down anything that wasn't immediately removed from the primary stage and allow it to be broken down further. So we can try to extract as much water out of it to make it usable as possible so we're not wasting any through the processing step. So biodegradation creates sludge. The sludge can then settle out, and then any leftover water can be processed later on. So those microbes actually break everything down as much as possible so that we can kind of separate out the usable water portion as opposed to the leftovers, the organic material and stuff like that. Then we go into the tertiary stage. The tertiary stage, we will filter out that water process that came from that sludge production. We'll filter all of that stuff and then also treat it with chlorination and possibly UV as well. This is not as clean as drinking water. So in that first process, this is a very basic, we will accept a little bit of dirtiness here, but it can be returned back to the ecosystem. So it's considered safe enough for watering golf courses and parks, but not for drinking at this point. But it can also be returned back to large bodies of water, which we can then put back through the more stringent filtering methods that we talked about with our drinking water, okay? So this could come back as drinking water, but it would be processed again before it came back, okay? The solid waste can also be used as fertilizer and applied to the land to help it recover and reintroduce nitrogens and things like that into the environment. So we try to eliminate as much waste of the waste as possible during this process. The most important part of wastewater management is basically managing microbes in that whole process. And this is wastewater surveillance, making sure that the harmful guys aren't making their way through. This was a major concern during the COVID-19 pandemic. Less and less now as we've learned more and more about the virus. So COVID is a disease caused by the virus, SARS-CoV-2. CoV stands for coronavirus. So SARS is severe acute respiratory syndrome, coronavirus number 2. SARS-CoV-1 caused SARS. If you weren't there for that, then you weren't there for that. Don't worry about it. It's okay. But SARS is different than COVID. They're different diseases. We'll talk about that in the future when we learn a little bit more about SARS and other respiratory diseases. COVID is a little bit more systemic and it caused a much more serious disease. There's a lot to be said about where we think it came from and why it is as severe as it is, which we will talk about in future chapters. But the virus, SARS-CoV-2, it's excreted in urine and feces, just like polio was. And so we had to have methods to get rid of polio back when it used to be prevalent in the United States that we didn't have to worry about once we thought we eliminated polio from this country. But we had to reintroduce those filtration methods and those screening methods after COVID came around because it was being secreted back into our water sources and then treating the waste and then treating back into drinking water. It wasn't enough to eliminate it from the system. And so there had to be more monitoring from that alone, let alone the normal microbes that you would see in waste, like E. coli and cholera and things like that. So the whole point of this is to make water drinkable, acceptable for consumption. And any clean looking, if the water looks clean and is safe, we call that potable water. Okay, so the potable water supply is that clean, healthy, hygienic water. It might not be entirely 100% pure, but it's safe enough. So safe enough water is called potable water. And so that's the whole point of all of these processes with the drinking water filtration and the sewage treatment is to create this safe and dependable source of potable water for the community. This means screening for things like Giardia, Cryptosporidium, Campylobacter, Salmonella, Shigella, Vibrio, Mycobacterium, Hep A, Norwalk, just to name a few, including COVID and polio and all those things. We have to make sure that these agents aren't making it through into our drinking water, so we have to monitor it. One of the best ways to monitor our water treatment system is through using indicator bacteria. Indicator bacteria are bacteria that are normally seen in the gut. They're normal in the gut. If the gut bacteria are found in your water, then the pathogens could be there too. We don't even need to test for the pathogens. If any gut bacteria made it through, then pathogens could make it through. So those are our coliform bacteria. We call them coliforms, fecal coliforms. If they are normal gut bacteria present in the water, then pathogens could make it through the process too. So this is just normal E. coli, Citrobacter, Klebsiella, and Enterobacter. They shouldn't be making you sick, but if these guys got through the water filtration process, the pathogens could too. Cholera and Salmonella and all of those guys could make it through too. So many of these are normal in your gut. They're gram-negative, fermenting, gas-producing bacteria. But if they make it through, the coliform bacteria, they are indicators that pathogens could too. So this is a major concept in our water surveillance. So we perform coliform assays, and we've been doing it since 1914. Basically, we use coliforms as an indicator that if these guys made it through, the bad guys can make it through. And there's a certain number that we would consider acceptable in certain water usages. So we would accept less in drinking water than we might accept for watering a golf course. And so how many are acceptable in your water sample at any time? It gives us some reference. And that's the importance of the fecal coliform assays. Some of our freshwater environments might have normal numbers of these guys. And now we've also started, looking at maybe E. coli alone might be a better indicator of fecal contamination as opposed to just environmental presence of microbes. And how important is it that we know that microbes are there versus the fecal guys? And the fecal guys are usually the indicator that fecal-borne diseases could get through. And so there's a little bit of discussion about that. But definitely our fecal coliforms are our main indicators that pathogens could be making it through. So how do we look at that in our water? Well, we quite literally filter the water to catch anything that's in it onto a little membrane and then put it onto an agar plate, like in a petri dish like we use in lab. And then we let it grow up and see how many colonies are there. And remember, for each colony, it was one cell. And then we would say that this has this number of fecal coliforms present. So we count both non-fecal and fecal colonies. So it's just the number of bacteria. And so then we might do further testing on any colonies to see what kind of bacteria they are or grow them on special media so that we can determine if they are fecal as opposed to just present in general. And so it's important that we know how many are fecal because, again, our fecal-borne disease-causing microbes are what we're really looking out for because of through all of this process, right? So again, we're going to be filtering our water through a filter, which we see here in this image on the left. Here, the water passes through the filter right here. And what comes out is filtered, and then all the microbes stay stuck on that filter that's in there. We then take that filter and put it onto a Petri dish and let it grow. And then the number of colonies, we can count them. And certain types of media and certain types of filters might change color if they're fecal guys or whatever. And so we can use this as an indicator of how many fecal coliforms are present in water when we filter it. Microbes aren't all bad, though. In fact, we often use microbes in our food to give extra flavor and texture and aromas like that. Kombucha is obviously a fermented beverage. We have pickled cabbage. Like when we talk about sauerkraut, that's pickled cabbage or fermented cabbage anyways. We have Norwegian fermented fish. We have Limburger cheese. Most cheeses, for example, we're going to have some fermentation going on in them. And we can even have, cultures like yogurts. We can have bread, which has yeast in it that ferment and create carbon dioxide gas with pockets of air in the bread and all of this stuff. So we often use microbes in our food. With food fermentation, we are putting it in a controlled environment to favor fermentation instead of growth through aerobic processes. So fermentations also tend to be synergistic. We're basically having more than one microbe often can work together to favor fermentation and production of those smells and aromas and the product of what we're looking for, like the rising bread and things like that. Bread microorganisms often are basically turning our bread into like nice fluffiness by producing the gases and helping with production of aromas that we associate with our food and things like that, and also helping conditioning the flour in order to make it workable and helping basically favor gluten production and things like that in the bread. So it gives the bread a little bit more elasticity. I bake bread, and so this is something that I have, you have to learn to work with the microbes that you're working with. Some of them are more forgiving than others. We see this a lot. If you eat bread, you have microbes to thank for making it nice and fluffy and spongy. We also have beer. This is basically the production of ethanol, which is alcohol, drinking alcohol, through fermentation. So we have certain microbes that, again, we force it into fermentation by eliminating oxygen presence. And that makes the microbes, often yeast and bacteria, produce ethyl alcohol, known as ethanol, which is drinking alcohol. It also produces carbon dioxide, which leads to the gassiness, the carbonation, that's what I'm trying to say, the carbonation naturally of the beer itself. So carbonation can be natural just from fermentation process. And that's also where we will get the alcohol from. Kombucha goes through a very similar process, but with less alcohol production. It has a light fizziness from natural carbonation, again, from the fermentation, from live cultures that are present in these areas. There's a bunch of steps that are involved in making beer, such as basically malting, where we would sprout the barley and then get the yeast that is available to break down like the starches and stuff that are available in the malts, in the barley. And then that we could then lead to making a mash to allow the enzymes that are involved in the microbes to be cooked out and stuff like that, and to favor the production of beer. And it's a multi-step process that leads to eventually the production of wort, which is essentially a filtered liquid from that whole mash process, where we would allow for inoculation with more yeast to favor the alcohol production after that. So then those yeasts would basically convert any of the sugars that were left over in that wort, the liquid portion of all of that barley, cooked barley and everything like that, to turn it into ethanol and carbon dioxide for our carbonation process. So that's the whole process for beer production in a nutshell. Next is the concept of One Health. So One Health is the idea that all life on Earth is connected, and microorganisms circulate among human hosts, animal hosts, and environmental reservoirs. We can see them, even one species can be in all three of those places. And so it's all interconnected, how they're interacting with our environment and with any of the living organisms in the environment and how they affect one another. And One Health is the idea that all of this stuff is interrelated, and the healthiness of the animals around us can lead to the healthiness of humans, and that maintaining the overall well being of our environment is key in keeping a healthy environment overall in our world. There's some huge challenges right now, especially relating to climate change. We have extreme weathers resulting from climate change, and we have record high and low temperatures. Temperatures aren't as stable or predictable as they once were, even in my lifetime. If you haven't talked to anybody who's older, like Oklahoma's climate was drastically different when I was a child. We had, snowy Christmases regularly, predictably. And nowadays, it's just changed. It's changed drastically. And there's different levels of allergens as we have longer warm days. We have rising sea levels from the melting ice caps. We have variable rainfall patterns as areas get warmer and it rains less, or some areas get warmer and it rains more, just depending. It's changing the ecosystems around us as a result of these weather pattern changes, from the changes as a result, many times, of these greenhouse gases causing warming around our planet and availability of food. But with rising temperatures in areas, we have rising mosquito populations too, and mosquitoes come with illnesses. And managing mosquito based illnesses is very critical, especially when we're talking about things like malaria, which are pretty prevalent in our tropical areas, around the center of the equator of the world. And more, we're seeing mosquito borne illnesses move north as the temperatures get warmer and warmer further and further north. Not just malaria, but diseases like dengue fever, chikungunya, and all sorts of other mosquito-borne diseases. And it includes West Nile virus and Zika too. So, how do we control all of that? Well, in areas where you're seeing increasing mosquito populations, the best way to control it is by controlling the mosquitoes themselves through use of nets, elimination of standing water, and using mosquito repellents and things like that. That's the best way to prevent the spread of the diseases. But other than that, like, it's pretty much, we can't control the climate at that point. So keeping a lookout and keeping vigilance on maintaining the mosquito populations is the best way to manage that for now. But that concludes chapter 25, and I will see you for the next one.